Hello everyone, in this video I'm going to talk about photosynthesis. But as you can see, we have already talked about photosynthesis in a not higher level, in a standard level. But this slide will include much more details related to higher level. As an essential idea, you know that light energy is converted into chemical energy. That's the whole process and the main reason for doing the photosynthesis. If you have a look at these two beautiful pictures, the left-right is photosystem 2 and the right side is photosystem 1. We are going to talk about the role of photosystem 2 and photosystem 1 later, but in brief you will notice that these are the protein complexes located in the thylakoid membrane, and they are absorbing the energy and they pass the energy by exciting the electrons in order to make the ATP at the end of the reaction. So let's move to the next slide. Well, there are understanding, applications, and skills that you are supposed to learn at the end of this slide. So since there are so many things, I'm not going to read. If you want, you can pause the video. But briefly, you will learn a little more details about the reactions. And the reactions includes light-dependent and light-independent, and also more details related to the names of the chemical reactions and inside the Calvin cycle. Those are more with details you will learn about them. So for the synthesis formula, starting with the carbon fixation, with the water molecule photolysis, with the usage of the sunlight, production of the glucose, and the oxygen. But we are not going to learn this stage based on this formula, but we are going to learn based on two different separate reactions, which are called light dependent and light independent. So let's move on for the first type of the reaction, which is related to light, and we call it light dependent. By the way, this is a great video, and you're going to watch this video by yourself, which you have the access by click on the link once you have the file. So let's have a look at the chloroplast structure in a TEM image, which is an electron micrograph picture. There is a chloroplast envelope which includes inner and outer membrane, and then you will see a stroma. The stroma is inside, and then includes the 70S ribosomes and the DNA. It's kind of similar to mitochondrial structure, a little similar, but with different names. For example, here we have stroma, and there we have matrix. And then here we have the thylakoid membranes, but there we had crystal. So almost same kind of a structure, but with different names and different functions. And in terms of thylakoid, there is one more difference between the mitochondria and the chloroplast, that thylakoids have the two kind of collections. Once when they are all just thylakoids, when we call it thylakoid, but then we have a stack of thylakoids. It's like a frisbee structures that they are stacking together. It's like this. So in that case, it is called granum, and granum is singular, and once we have the multiple granomes, just this granome, granome, all of these that they look like stacks together. Each of them is called granome, and the granome, when they are together, then that would be grana as a plural form. The important part that which structure is doing what is that you remember that the granome side is for light dependent reaction, because those electron transport chain, which is similar to mitochondria, is located, the protein complex is located in the thylakoid membrane, and these are the structure that includes the thylakoid membrane. But once the light dependent reaction is done, then that will be the light independent, in which those energy that has been transferred to the molecules will be used for production of the glucose, and this happens in the stroma. Let's move to the next slide. So as I mentioned, we have two separate reactions divided into light dependent and light independent. Once we are talking about light dependent, then there are some reactions happening only in the presence of light, and some reactions happening when the light is not necessary. Okay, with the light dependent reactions, once the light is necessary, what happens is that the NADP plus and ADP will finally will turn into or converted into ATP by phosphorylation. This is called photophosphorylation because it's with the usage of the light. If you go back to the mitochondria, then you remember it was oxidative phosphorylation, because the phosphorylation was got the energy from the breaking down the glucose, but here is the energy comes from the light, so that's why it is called photophosphorylation. And then one more thing is that in the photosynthesis we have NADP plus. It's not NAD plus. There is one more P added to the molecule, and you can remember P for photosynthesis, so that's the way that you remember that we have NADP, not NAD. And if you want to memorize in a better way that you don't forget, you can look at the idea that, okay, we need light in light dependent, that's easy, but what else do we need? We also know that when there is a light, this means that we want to see how the energy comes from the light will be used or will be stored in kind of molecules. So what kind of molecule store the light energy are ADP and NADP. So we need ADP and NADP in the light dependent. And once we are thinking about NADP and ADP, then who is going to give them the electron? Then that would be water molecules. So water molecules will be broken down, which is called photolysis. And by broken down, it release the oxygen molecules, and at the same time it gives the hydrogen protons plus electrons both at the same time to the NADP plus and ADP. So in that case it becomes ATP, which has already reduced or got the electron, and NADPH got the proton and electron at the same time. So this is how you remember. Once again, light-dependent reactions needs light. We need some molecules in order to get the energy from the light, which are ADP and NADP plus. And we need something for giving the electron to this molecule, which is water. So water and ADP and light, all of them are necessary for light-dependent reactions. Where does this happen? It happens in the thylakoid membrane space. With more details, this structure, which is located in the thylakoid membrane, and it explains the light-dependent reactions. The same as mitochondria, if you remember that we had protein complexes, but in mitochondria we had protein complex 1, 2, 3, and something here as 4 at the end. But in Chloroplast, it's not the numbers based on the order, because it starts from photosystem as a protein complex, photosystem 2, and then there is one more structure here, and then we have photosystem 1, and we have ferredoxin over that you need to learn from the thylakoid membrane protein complexes is mainly photosystem 2 and photosystem 1. Why is it photosystem 2 at the first and then photosystem 1 here? Well, it doesn't, the name is not because of the order, is because of discovery. So photosystem 1 was discovered first, but the location is here. But because it was discovered first, so that's why it is called photosystem 1. And photosystem 2 was discovered later, but the location is here. But anyway, the light-dependent reaction starts from absorbing the energy by photosystem 2 here. Play this video and watch together later. So the chlorophyll in photosystem 2 inside the thylakoid membrane absorb the energy, and then electron get excited, and then this electron excited will be passed to the next protein complex, which is here. And after passing the electron, after getting excited and passed to the next protein complex, then you will observe that here is the stroma and here is the thylakoid space, which is inside the thylakoid, and the electron comes from the photolysis of the water molecules by breaking down to hydrogen protons and oxygen molecule. And once the electron will be passed to the different protein complexes, the final stage would be ATP synthesis, exactly the same as mitochondria. It is called chemiosmosis, the same thing, because the protein complex is the same as ATP synthase. The function is making ATP, and the energy comes from the hydrogen protons gradient, because the water molecule here is breaking down into proton ions and oxygen molecule, and the increased level of the hydrogen protons make the gradient that will be used for as a potential energy for usage of the ATP synthase. And again, another animation that you can use from this link. And here we go, that the hydrogen protons will be used for the gradient and for production of the gradient, and then the ATP will be produced after conversion of the ADP to ATP. One more thing, that when does NADPH produce? Well, that's the function relates to ferredoxin after receiving the electron excited by the photosystem 1. So the photosystem 2 received the light energy and then passed the excited electron to photosystem 1. And again, photosystem 1 received the energy. So both of them, both of the photosystem 2 and photosystem 1 received the energy from the light and use that energy for exciting the electrons. But photosystem 1, after exciting the electrons, it passes the electron to another molecule which is called ferredoxin, and this ferredoxin is the function is that passing the electron to the NADP, and so far NADP plus protons, because the protons are already here by the usage of the excited electron, then the NADPH will be produced. Almost the same process as mitochondria, that NADH and ATP was the production of the respiration in the electron transport chain. This is a review from previous unit, unit 2.9, related to action spectrum and absorption spectrum. If you want to review, you can go back and watch the video or the slides in the past. So in summary, we have the absorption spectrum, this one, the black graph showing the absorption, and as you can see, the absorption is maximum in like 680 and 700 nanometers at this peak, and this peak, which is in the red color, and it has the maximum rate of the absorption at this stage, at this area, and almost this area. So one more thing that you will learn here, which is additional to the previous slide, is the range of the absorption spectrum for the chlorophyll A and B, and this is what you need to learn and remember. Chlorophyll A is having a wider absorption spectrum, which means that starting from before 480 at this stage, which is like something like 430, and after, which is something like near 700 nanometer, and chlorophyll B is inside the range of chlorophyll A. So chlorophyll A, you can remember A is the first one, so it's having the wider range, and chlorophyll B is the second one, is inside the range of the chlorophyll A, and this could be like something like this. And this is the absorption spectrum for two types of the chlorophyll A and B.
In plants, photosynthesis occurs in specialized organelles called chloroplasts. The internal membranes of chloroplasts are organized into sacs called thylakoids. Surrounding the thylakoid membrane system is a semi-liquid substance called stroma. Photosynthetic pigments are clustered together to form photosystems. When a photon of light strikes the reaction center of photosystem 2, it excites an electron. Two water molecules bind to an enzyme at the reaction center. This enzyme splits the water and uses the electrons from the water to replace the electrons removed from the reaction center. Oxygen is produced in this process. The primary electron acceptor for the light-energized electrons leaving photosystem 2 is plastoquinone. The reduced plastoquinone passes the excited electrons to a proton pump embedded in the membrane called the B6F complex. Arrival of the energetic electrons causes the B6F complex to pump protons from the stroma into the thylakoid space, thereby generating a proton gradient across the membrane. Because the thylakoid membrane is impermeable to protons, the protons in the stroma must pass through the channels provided by ATP synthase. As protons pass through, ADP is phosphorylated to ATP and released into the stroma. This process for making ATP is referred to as photophosphorylation. When photosystem 1 absorbs a photon of light, its reaction center passes high-energy electrons to ferredoxin. The enzyme NADP reductase then transfers the electrons to NADP to form NADPH. Electrons lost from photosystem 1 are replaced by electrons generated from photosystem 2. A small protein called plastocyanin, PC, then carries the electrons from the B6F complex to photosystem 1.